Hi everyone, my name is Lindley and today I will be explaining how forces interact to stabilize the double-stranded DNA. To preface, it is important that you have a solid understanding of the nucleotides responsible for making up DNA. These nucleotides are made up of the base, the phosphate group, the sugar, and attached together by one of the various ribose carbons that compose the sugar molecule. The 1' carbon attaches to the bases. The 2' carbon distinguishes DNA from RNA and binds to a hydrogen. The 3' carbon bonds to the phosphate, yielding the formation of the phosphate backbone, and the 5' carbon is connected to the phosphate group. There are 4 bases that can be seen on these nucleotides. They fall under 2 categories: purines and pyrimidines. Purines include adenine and guanine and can be distinguished by their 2 rings. Pyrimidines consist of cytosine and thymine and can be identified with only having 1 ring. Base pairing is how these bases pair up. Base pairing in this case happens when adenine and thymine bind together through pretty weak hydrogen bonds. Cytosine and guanine also bind together by way of weak hydrogen bonding. Since these base pairs are all hydrophobic, strong hydrophobic interactions are utilized so that the DNA strands are kept together. These interactions include base stacking and the phosphodiester bonds created, which are used to strengthen the interactions within the DNA double helix. Base stacking happens when bases of molecules stack up on another. Here you can see this happening within the purines and pyrimidines. You can also better see the phosphodiester bonds here too, as they bond a sugar to a phosphate. This is the DNA double helix. As the labels indicate, it is composed of 2 negative phosphate backbones which are hydrophilic, and the base pairs are stacked within the structure. It is common for helix structures to have handedness, meaning it favors toward the right or left hand. Taking a closer look at this DNA double helix model, the overall structure favors the right hand side, giving the structure its right-handedness, which is the most common type. You should also know that the 2 strands on either end of the structure are considered antiparallel to one another. We know these are antiparallel strands based on the direction in which the nucleotides are connected. One strand reads from 3' to 5', while the other strand reads 5' to 3'. Something else to note about this double helix structure is that the spaces within are not evenly distributed, which creates for major and minor grooves within the DNA. Major and minor grooves are significant because minor grooves have less space, while major grooves have a wider space among sections of the structure. Minor grooves create less space for proteins to engage with base pairs compared to the major grooves, which have a larger amount of space to interact. Going back to the negative phosphate backbone, this charge on the double helix structure creates a force that is destabilizing for DNA and impacts the midpoint of thermal denaturation, or TM. Right now, this causes a lower TM for DNA, but adding a salt-concentrated buffer can help to better stabilize the initial force and increase the stability of TM on DNA. This action would make the TM higher, as an increased amount of energy must exist in order for the strands to move apart. Ultimately, what has happened by adding the salt buffer is the neutralization of the previously negative strand, making it more stable as seen through the tighter, stronger bond above. I hope you have found this video informative and helpful. Thanks for watching.